Hi, everybody. Welcome back to the UCLA CART Distinguished Lecture Series. This is a lecture series that happens once a month where we bring in our favorite, excellent, great guest speakers to provide you with the latest scientific updates about autism. This lecture series is sponsored by the UCLA Tarjan Center, the UCLA IDDRC, the UCLA Brain Research Institute, and the UCLA CAN Research Training Program. So I will introduce our speaker. You will be able to ask questions via Q&A after the talk, and you guys in the audience can ask them in person to the live person. We are also recording this talk, so you will have it available on the website. So Dr. Blair Braden is an associate professor at Arizona State University. And she is a completely unique person because she is really one of the major leaders, if not the major leaders, in actually doing neuroscience that addresses aging in autism. And as we all know, people with autism get older. And we know almost nothing about what happens after sort of early adulthood in terms of systematic research. Even more so, what we do know is mostly from large-scale studies of medical records or Medicaid data. And so what Blair has been able to do has been able actually to carry out neuroimaging and actually some treatment studies with an aging population. She comes to us as with a bachelor's degree from University of Kentucky in psychology and mathematics, and then went to graduate school at ASU and managed to stay on, do a postdoc at the Barrow Neurological Institute, and then stay on as faculty and as now an associate professor. So we're all really looking forward to hearing what she's got to say and supporting her throughout her career to figure out what goes on in aging and autism. Thanks, Blair. Thanks for coming.
Thank you so much. It's wonderful to be here and to talk about aging with autism. Okay, so I think of my lab as having these two prongs. The first and foremost thing we do is a longitudinal observational study to just see what happens to autistic adults as they grow older, and really with a heavy focus on cognition and brain. Then when we find things out that are persistent or worsening challenges, we try to develop support programs that can they can help improve the lives of autistic adults as they grow older. So you all probably don't need this timeline, but I'm going to do it anyway. Oftentimes when I'm talking to people who know nothing about autism, it's so helpful for them to see when was autism introduced back in the 40s, that it took almost 40 years before it was really a diagnosable condition from the DSM, that it went from a rare condition to one of the most prevalent neurodevelopmental conditions. One anecdote I like to tell is I have a professor who I work with who's a speech language pathologist. And when she went to school, she had a half day on autism, and she was told, this is rare and you're probably never going to see it in your career. And then most of you are probably familiar with Donald Triplett, case number 1 in Leo Kanner's original study, and he just passed away this last year. So before, this was the first time I updated the talk to say that, because before it was, he turned 89 this year, he turned 88 last year. So it really helps set the context of how can we know much about aging with autism when the first kiddo to ever be called autistic was still alive until this year. So how I came into this field was not from an autism lens, was not from a developmental lens, was from an aging neuroscience lens. And got the opportunity to, or sort of the invitation to work in this field. And there were these kind of publications were really the only thing that was out. We should be doing work in aging with autism, but there was very little actually being done. So it was wonderful to have papers like this to sort of guide me as a roadmap for what needs to be addressed in this field and how could I bring my skills as an aging neuroscience researcher to help answer some questions. And then when I started meeting with autistic people and talking to autistic people, wow, is this one of the things they're most afraid of. Autistic people themselves afraid of growing older because they don't feel like the healthcare and larger community knows how to best care for them. Parents who are scared of their kids growing older and what's going to happen to them when they're not around to take care of them because people don't know how to best take care of them. And then 2 years ago, so I had been doing this work for 6-7 years when this article came out, was a real, when we started this work, we didn't really know if there were going to be problems with aging and autism that needed to be addressed. And when this article came out, it really kind of galvanized us again for what we're doing and why we're doing it. And that, yeah, there probably are some unique vulnerabilities, in particular, 2.6 times more likely to be diagnosed with an early onset Alzheimer's or other dementia. So some of the things we're going to talk about today are first, a few of the major observational cognitive and brain aging findings we've had so far. So it's going to focus on executive function, memory, and the hippocampus as kind of the main things that come to light. Sex differences is a big focus of what we do. Sex differences is important in aging, super important in autism. You put them together and there's a lot to figure out. Starting to get into some understanding genetic contributors to aging and autism risk. And then the goal of, what I would like to produce in my career, and so many other neuroimagers feel the same, is can we actually use MRI to help inform healthcare? Can we use MRI to predict who's most at risk for accelerated decline? Parkinsonism is a big thing that has come out of the aging and autism literature. So we've started to dive into that as well. And then a couple of other aging observations I'll mention are findings in mental health and social communication challenges. And all of that will hopefully set the stage for the support programs that we've been working on and how we're trying to address these problems. So the very first thing we set out to do was really just determine if cognitive and brain differences that we see in younger autistic people are even still present in later life, or have things gotten better and there's really no difference between middle-aged and older autistic adults. That really was unknown when we started. So in a very small sample of 40 to 7-year-old men, also in the beginning, we were like, it's going to be so hard to find middle-aged and older autistic people. It's going to be really hard to find women. We know sex is a big factor in both autism and aging. We need to start with a homogeneous sample and start with men, which is like a horrible thing for me as a researcher to have spent a couple years telling autistic women I couldn't include them in my study. That changed. So executive function was one of the things we first looked at, some of the most prominent cognitive findings in younger people with autism. Indeed, in our middle-aged and older autistic adults on the Wisconsin card sorting task, we saw more errors compared to our neurotypical controls. In the brain, one of the big findings that stood out was smaller hippocampi bilaterally. So this is our memory brain structure. And then looking at the white matter through diffusion tensor imaging, fractional anisotropy is a measure that roughly evaluates the integrity of the white matter fibers, so the connections between gray matter regions. And the connections going into the hippocampus showed some signs of lacking integrity compared to our middle-aged and older neurotypical participants. So that set the stage for what we need to follow up on. And this is a demographic table of where we're at right now. So the baseline, we have about 123 people who are over the age of 40 in our study, roughly half autistic and half not autistic. Baseline age is 40 to 70, but we'll take people as old as we can find them. We have about a 2-to-1 male to female ratio in the study, which we work very hard for. And we've gotten at least one time point of follow-up data on 90 of these folks. Some people we've been following for 8 years, and we try to get a new data collection every 2 years with our participants. This study does not yet include individuals with intellectual disability, but I hope to change that soon. So you can see we have an average and comparable range of IQ. So pictorially, this is what our data looks like or looked like last year, with all of our participants on the y-axis and age on the x-axis. Each dot is a person. And then as the dots go out to the right, that's a data collection point that we have on them. So we have a young adult cohort, 18 to 39, that we've collected one time point of data. And you'll hear me talk about this for cross-sectional age relationships. And then anyone over 40, we try to get them to come back every 2 years to look at age-related changes. And we hope in a few years, our data will look like this, but I really hope even taller, actually, because we're at this stage trying to make a really concerted effort at improving diversity in our sample. It's hard to find middle-aged and older autistic people. It's even harder to find diverse middle-aged and older autistic people, but we're really trying to get into the minority communities in Phoenix and change that. So the first piece of longitudinal data that we had was, or kind of difference, was in verbal short-term memory. So this is the auditory verbal learning test, the AVLT, the A1. The first time someone reads 15 words to you, how many can you remember right off the bat? And if you do this with someone twice, you can calculate something called a reliable change index score. And that is the difference between your performance in these two times within the margin of error, or is it so different that we can say this is either clinically meaningful improvement if we're trying to improve your memory, or clinically meaningful decline, which is, you know, the memory improvements are rare at this age. So what we found, looking at the data kind of in a categorical chi-squared way, is that our middle-aged and older autistic adults were significantly more likely to fall in that clinically meaningful decline category than our neurotypical adults. And this, you can see, is not very many people. So this was sort of surprising to have this kind of chunk of people after only 2 to 3 years really go down on this test. However, it should be noted that the vast majority of people are stable. So although this was a surprising finding to us as really showing some potential risk, it's also very clear that aging with autism is not a deterministic experience of a major decline in cognition, and that some people will probably decline faster, but lots of people won't. In the brain, we saw structural differences that went right along with this. So this is the hippocampus, that part of the brain that's important for making new memories. The age over time is on the x-axis. The size of the hippocampus is on the y-axis. The hippocampus gets smaller as we age. The hippocampus gets a lot smaller in Alzheimer's disease. At this middle age range, our neurotypical adults' hippocampi aren't really changing over 2 to 3 years. But on average, our middle-aged and older autistic participants' hippocampi is significantly shrinking. We have added a third time point to our data, and we see very similar trends. So this is long-term visual memory, where across time on the x-axis, our middle-aged and older neurotypical adults are showing very stable performance on this test after 2, 3, 4, 5 years of time. And our autistic participants are showing an average rate of decline over this 2 to 5 year window. We have some unpublished, very preliminary sex differences in longitudinal memory change. So this is now verbal long-term memory, the AVLT delayed recall measure. One thing that you'll note is the men's lines are much longer because we started working with them about 2 to 3 years before we got funding to recruit women as well. So we need to take this with a grain of salt, right? Anytime you followed a group of people for longer, you're more likely to see decline than when you follow someone for not as long. However, the trajectories are fairly compelling, and even with a small sample size, this is a significant sex by diagnosis group by age interaction. The blue line is our autistic men that's showing a very clear downward progression, and everybody else is pretty stable. So the autistic women are in green there, and they at this stage really don't show any signs of decline as a group collectively. So as someone who comes from the field of aging and thinking about genetics, ApoE is the gene that we talk about all the time. So this is the strongest risk factor for Alzheimer's disease. In the world of polygenic disease and conditions, ApoE is a very rare thing in how prevalent the diversity of the APO gene is and how strongly one of the alleles contributes to Alzheimer's risk. So I was surprised to find there's actually a rather older study at this day and age that had looked at APO alleles in kids with autism and found that the kids were more likely to carry the high risk E4 allele. We looked at it, and our sample at this point was probably only about 70 or 80 participants and found the same thing. So that was rather shocking. And of course, we needed to see if it was influencing the cognitive and brain presentation of our participants. So we just published this paper that's a very preliminary look into how this gene might be playing a role. The AVLT, again, was the measure we're using. And We only had a few females who we were able to genotype that were contributing a lot of variants, which again, there's sex differences in Alzheimer's and the way E4 affects memory and learning. So when we removed our women from the analysis, this E4 high risk allele was only negatively affecting a cognitive measure in our autistic men. So our neurotypical men and neurotypical women in this middle age range did not show a measurable effect of this risk allele, but our autistic men did. And then the first sort of hint we have towards could we possibly predict who's most at risk to decline in memory based on imaging is centering around the hippocampus. So the hippocampus is our memory structure. If people are going to decline in memory, can we see it here? Can we see the early signs of it here? Hippocampal volume, which is what I've talked about thus far, is classic, just the size of the structure, and it correlates with people's memory abilities. But there is another type of imaging that's a bit newer and perhaps more sensitive that you can do through diffusion measures. So diffusion tensor imaging, or DTI. People typically use this type of imaging to look at white matter. So this measures how water moves around in the brain. Water slides right down white matter tracks and makes really cool images of how our brain's connected. But you can also see how water ping-pongs around within gray matter structures. So if you have a gray matter structure that's packed very densely with neurons and synaptic connections, water doesn't have much room to bounce around. If you have neuronal loss, synaptic loss, water has more room to move freely, and we can measure that. So I have wonderful colleagues who have really pioneered using this technique in aging fields in Alzheimer's and Parkinson's, and show that you can really look at microdegeneration with this technique that is more sensitive to functional decline. So we compared these two measures head to head in our data, and also it was the free water that came out as correlating with future memory decline. So when we measure how much free water does someone have in their hippocampus, the participants that had the most free water then went on to show the greatest memory decline in the next 2 to 5 years. And this is specific to the autistic group. So we have high free water on the right of the x-axis, and then the y-axis is memory change, so that those negative numbers mean a memory decline. And it's very clear over a certain point of free water, you're in the decline category. So while we're over here chugging away, thinking about cognition and memory, which is what, really jazzes me up, the world of aging and autism kept coming out with these papers. This field is small. There's not a lot of publications. There's not a lot of cohorts. So in a small body of literature, when there's 4 independent studies, largely assuming, that are showing the same clinically pretty significant change, it was something that we needed to investigate. So this last year, we've been calling our participants, emailing them, getting them on Zoom, and asking them to just answer a really simple 8 questionnaire about their experience of Parkinsonism in their life. So we have gotten in touch with about 75 or so of our participants who are over the age of 40, about 30 some autistic, 30 some non-autistic. And I was absolutely shocked to see how strong the effect, actually, this is not over 40, this is 18 and over, how strong the effect was in self-report Parkinsonism, where our autistic participants are reporting way more of these symptoms than our neurotypical participants are.
How many people is this?
So this is about 35 in each group or so. So my next thought is, can we see this in the brain? Let's look at some memory brain structures, which is not what I typically am drawn to in the brain, but with a behavioral finding this intense, I was very motivated. So one brain measure that supports the memory system that popped out to us is the caudate. So this is part of the basal ganglia that's involved in motor planning and execution. And looking into the literature, there are several studies that show that autistic people have larger caudates, and this is thought to be tied to restricted repetitive behaviors. We showed this as well in our participants that were more in the young adult range. However, if you follow these age relationships, the red line is the autism group, age is on the x-axis, size of the caudate is on the y-axis, the red line has a steeper negative slope. The autistic group has a steeper negative slope. So potentially, although autistic people may start off with larger caudates, there could be a greater age-related decline in this motor brain structure. So at this stage in analysis, this group by age interaction comes out at a 0.07 significance level. If this does end up being the case, that older autistic adults have smaller caudate, that would be similar to what is seen in Parkinson's disease patients compared to healthy controls. Some of the other brain regions we've looked at and have not seen any differences are the precentral gyrus, the primary motor cortex, the cerebellum, which does a lot of motor adjustment roles for our brain, and then the putamen, another striatal structure. So those things are sort of similarities between autistic people and people with Parkinson's, but they're even more interesting are the differences. So first of all, the caudate, the size of the caudate, which is on the x-axis, has no relationship with whether or not the person is reporting Parkinsonism. And this is different from what we see in people with Parkinson's disease. If you have Parkinson's disease, the smaller your caudate is, the more severe your symptoms are. Even more shocking, there's no relationship with age and Parkinson's symptoms in our autistic participants. So we went back to our participants as young as 18, and youngins are showing Parkinsonism to the same degree as our middle-aged and older ones are. And if you look down there, so we have, age on the x-axis again, severity of symptoms on the y-axis. Our blue line is our neurotypical group. They have very low symptoms, but even with very low symptoms, their age correlation is approaching significance, because none of our young adults who are neurotypical are endorsing symptoms for the most part. But when you get up to 55, 60, 70, you see a couple of symptoms. So there's a lot to do in this field. I mean, first we need to look at longitudinal change and see if this is something that is a part of some autistic adults' lives and doesn't really change, or is this something that's getting worse over time? One of the healthcare record studies that came out found that autistic women were more likely to be diagnosed with Parkinson's, so we need to look at that in our study. And then get even deeper into the brain imaging. So I mentioned free water and that it's been used in Parkinson's. This is my colleague, Ed Ofori, who's really pioneered this concept. What Parkinson's disease is loss of dopamine producing neurons in the brainstem, part of the brain called substantia nigra. Very, very tiny structure, but you can go in there and hand trace it and look at the free water. And people with Parkinson's disease, as you would expect, have way more free water because they've lost the neurons. So we're starting to do this in our autistic participants, seeing some very preliminary evidence that there is more free water in this substantia nigra brain region, suggesting there might actually be real pathological overlap or risk for Parkinson's disease in autistic adults. One of the great things about this is that MRI is non-invasive, and the other way that people look at neurotransmitters in the brain is PET and SPECT. So you can use these tools to even help diagnose Parkinson's. However, we can get a really good picture with MRI before we go to these tools. And then if we find evidence that suggests there are dopaminergic changes, then we'll follow up with PET and SPECT to look at dopamine. Mental health is something you absolutely cannot ignore when you work with autistic adults. In the whole adult autism literature, reduced quality of life, mental health challenges is probably one of the most consistent published findings. And we've published this in our cohort as well. When looking at mental health related quality of life and physical health related quality of life, autistic men and women report lower mental health quality of life. Only autistic women report lower physical health related quality of life. An interesting point of resilience in aging with autism is that when we look across our whole cross-sectional cohort from 18 to 71 or 2, and we look at the correlation between age and mental health challenges or mental health quality of life, we see that older autistic women are actually doing quite well with their mental health. So this is the blue line up here, that our older autistic women report high levels of mental health quality of life, and that those that are reporting the lowest levels are young autistic women. So when we get into talking about support groups, I'm really into the idea of intergenerational support groups for autistic adults, and the idea of older autistic adults being able to mentor younger autistic adults in these incredible skills of resiliency that they've developed over the course of their life. We can also take a deep dive into cohort effects in autism research, and also talk about, what are the sampling bias differences between someone who's 60 or 70 and participates in my study and someone who's 20 as well. Okay, lastly, social communication. This is not my area of expertise. It's not even particularly the thing I'm most interested in, but it's absolutely important in autism. I work in a speech and hearing area with a lot of SLPs, so I have a lot of great colleagues to collaborate with this, and I had my first PhD student, Melissa Walsh, here is one of those. So she took a peek at our SRS data, social responsiveness scale data, back when we were only working with autistic men, and found that our middle-aged and older autistic men were reporting greater challenges in the cognitive aspects of social communication compared to our young adult men. Her theory was this could be related to the executive function challenges that our autistic men are experiencing, that we use our executive function a lot to have effective social interactions. And if middle-aged and older autistic men are struggling with their executive function, could that then interfere with their ability to have successful social interactions? So in the brain, you can get a picture of kind of the brain basis of executive functioning through one of these resting state networks that includes the frontal lobes and the parietal lobes. So this has a lot brains on here, so I'm going to walk you through slowly. When we look at functional connectivity, so this is how different parts of the brain are talking together, and we expect that networks that are coordinated would be talking together in sync, and that we can measure that across different participants. And so we've like plucked out these two parts of the frontal lobe, the left and right dorsolateral prefrontal cortex. And when we look at how well these parts of the brain are talking to the rest of the executive function network, we plot them over here in these squares. So our neurotypical participants are in blue up at the top, young adult on the left, middle age on the right, high connectivity within these brain regions. Our autistic participants are in yellow, young adult on the left, a little bit lower than the neurotypical, but not too far. And then the right are middle-aged and older autistic adults who have far less connectivity in this part of the brain to the rest of the executive function network. Which in and of itself is, okay, that not to compel or not, blow you out of the water of things that we don't already know about neuroscience and autism. But what blew me out of the water is this functional connectivity correlated with those self-report social communication challenges. So our middle-aged and older autistic adults who are having the most challenges in social communication have the least connectivity in this part of the brain. And then to bring it all around, this correlation is also true for behavioral measures of executive functioning. So one executive function task, the Tower of London, that involves planning in order to not make mistakes. The autistic adults who made the most mistakes are the ones that are saying they have the most challenges with social communication. We have not followed these particular analyses up since we have recruited a sizable number of women in our study, but we did sort of slip in some age-related sex differences in social communication in a review paper that we did about sex differences in MRI measures of autism. So we were building off this study to the left by Wagner et al. That looked at longitudinal trajectories in SRS by a mother reporting on their child. And their finding that they were interested in is that when you compare the red line, which is autistic boys, to the blue line, which is autistic girls, around the age of puberty, the autistic girls sort of get a little better in their social communication challenges, and the boys don't. So we wanted to follow that up across the rest of the lifespan. We combined our data with data available through NIMH, same colors here. So red is autistic men, blue is autistic women. And the really big takeaway is there's all this variability in autistic girls and women across their lifespan. And I have a strong background in hormone research, so that's what I think about. Like, how are hormones influencing the experiences of autistic women, girls, and specifically throughout their life that differs from the experience of boys and men. And when we get to this age bracket, 50 to 60, a big thing called menopause happens that has a large effect on many women. And there's a small body of literature starting to come out about the really unique challenges that autistic women go through during the period of menopause. So those are our highlights. That's 8 years of things that have sort of bubbled to the surface. And I'm going to try to recap them in a couple slides. And when I was putting this together, it really occurred to me that what we have seen is both things that are common struggles with aging that have unique effects in autistic individuals, and then also common struggles with autism that have unique effects associated with aging. So first, in thinking about what we know is affected in aging and how that might be uniquely affected in autistic adults, learning and memory is one that has really risen to the surface. We honestly expected executive function to be the larger sort of area being hit by aging, and we're surprised that it really is memory. So there is some risk for accelerated decline in both short and long-term memory. Men may be more vulnerable in this decline, but we have a lot of work to really find out. And an Alzheimer's risk gene could play a role in why autistic people might show greater declines in some of these abilities. This is probably largely tied to the hippocampus, and that there is some risk for accelerated volume loss of this structure, and that it's possible that with advanced imaging of this structure, we might be able to predict who's at greatest risk for decline. Parkinsonism has proven to be something to really be concerned about with aging autistic adults, and that it's very interesting to see that young autistic adults report high levels of these symptoms. So there's a lot to figure out about, is this Parkinson's disease? Is this motor features of autism and how they're presenting in adulthood? And is it a little bit of both? And then tied to the brain, the caudate does seem to be different in autism, could be aging differently as well, but may not be a big part of the Parkinsonism phenomenon that we see. So now on the other side, things that are much bigger struggles in the autism community, as opposed to being huge vulnerabilities to aging, we see that mental health is persistently a challenge, with some signs that autistic older women may have developed some really great resiliency skills we can learn from. Social communication is a persistent challenge. Some men may struggle with this as they grow older, and some women may experience much more lifetime variability that could be related to hormonal changes. And executive function seems to be a persistent challenge, but not necessarily strong evidence that there's accelerated decline in these cognitive domains. Brain connectivity underlying these cognitive domains might be declining with age. We have to follow longitudinally to see, and but however, it does seem clear that executive function abilities is very tied to social abilities for autistic adults. So although we're very proud of our study that we've worked very hard on for many years, it's still small. We're still talking about less than 100 people in each group, and there's so much more work to be done with large cohorts of these age ranges and really seeing what effects are strong and reproducible. It's also worth mentioning that not all aging and autism studies find similar results. So Hilde Geertz is really the pioneer of this field, working out of the Netherlands. And although she shows persistent challenges in her middle-aged and older autistic adults, she doesn't have a whole lot of evidence for things that are declining faster. There's a lot of differences in her population versus some of the US-based populations. Her populations are entirely adult diagnosed, and ours are not. Ours include people that were diagnosed as kids. And then I think a lot about the different health factors of being someone who's growing older in the Netherlands versus someone who's growing older in Arizona or North Carolina or lots of other places in the US that just don't have the supports that some of our European friends have. There's so much more work to be done in genetics. We're barely scratching the surface. We will have whole genome data soon, so we can look at a lot of different genetic influences. I'm very lucky to be married to a neurogeneticist, so she and I do this work together. Some of the other noteworthy things besides just looking at inherited genes is telomere length is one thing we've published on in the past, which if you're not familiar, each of our strands of DNA are sort of capped with protective nucleotides. And the older we get and the more stress we're under, the more those get chopped down and make our DNA vulnerable to mutations. So we've shown autistic kids have shorter telomeres compared to neurotypical kids. That could put them at risk for other age-related vulnerabilities. And then what she really does is epigenetics. She does epigenetics in terms of kids and early life trauma, as well as psychedelic treatments, which is very cool. People love to talk about that. But there's also really exciting findings in the world of autism and epigenetics and aging and epigenetics. So we hope to get into that one day. And in general, so much data, so little time. I've showed a fraction of the brain imaging that we do. And you know, there's so much more to look at as far as different tasks people are doing in the scanner, different types of scans that look at different insults to the brain that can affect cognition. And then sex differences needs to be looked at for everything. How is age of diagnosis playing into outcomes, different levels of symptom severities, whether you have comorbidities, are you employed, are you not employed, what kind of medications are you on, which is particularly important in Parkinsonism, how are you interacting with the healthcare system, what supports do you have in life, what supports have you had previously, hormones, sleep, I could go on and on, which is where big data really needs to come in to be able to answer these questions. And then lastly, as I alluded to, we do not include adults with intellectual disability now, and I'm really tired of that and ready to change that. So I'm meeting more and more with people who do aging and intellectual disability populations and hoping to start recruiting autistic adults who have ID as well as people who have ID and aren't autistic. Okay, so all of that hopefully sets the stage for what things do we need to address. Memory and executive function, cognition broadly, mental health, and social communications are the things that really rise to the top of what we want to try to support. And frankly, mental health is the easiest one, I think, to tackle in that list. Mindfulness-based stress reduction is a program that I got some experience with during my postdoc. At this point, almost everyone is familiar with mindfulness. The broad theme of mindfulness is that you want to teach people adaptive coping skills so they can redirect their focus to the present moment and with a non-judgmental attitude. MBSR particularly is a 8-week-long course where you meet once a week for about 2 hours. It has been widely studied in clinical and non-clinical populations and is extremely effective. There was one group, again in the Netherlands, not related to Hildegard's, that had been doing MBSR in autistic adults. So we wanted to try that as well. We did not want to take the waitlist control approach because we know social support can be so impactful for autistic individuals. So that was one thing that we wanted to do differently. And then we did brain imaging before and after the mindfulness training, which I will probably never do brain imaging in a clinical trial ever again. It's so much work. So in our initial cohort, we found what others have shown, which is depression reductions associated with mindfulness. So this is each dot is a person, their pre-score, their post-score. The mindfulness is in green. The control group, which we called social support with relaxation education, is in orange. This is the BDI, so I have the different levels of severity in dotted lines, which is my favorite way to look at the data, because you can really appreciate that like people got better in their lives. This is not just mean scores of 1.5 points that reach significance. People largely drop down at least one level of clinical significance. The support education group, a lot of people got better too, but there was more variability with the response. As a quick note, we did find some cool brain changes. And in particular, we had people in the scanner making judgments about themselves, which if we're teaching people to be non-judgmental, you should change the way you think about yourself before and after going through MBSR. And we found that our participants going through MBSR sort of increased one of the parts of their brain, their anterior, their middle cingulate, when they were thinking about themselves. And the degree to which they started using that brain structure more in self-reflection correlated with how much better they got with depression. So that was our first rather small sample. We did this 2 more times to get a larger sample. and eventually ended up finding that our support group with relaxation education did just as well for reducing depression and anxiety, which is awesome, right? Like anybody can print off the NIH materials about relaxation and get a group of autistic people together and talk about stress and share strategies that are working for them. This suggests if those are the problems you're trying to address, you don't particularly need mindfulness. However, we did see that mindfulness expectedly improves mindfulness traits. And a little kind of hint towards maybe has some cognitive benefits as well, which has been shown in lots of other populations that mindfulness improves attention and other aspects of executive functioning. From a sex differences perspective, it was pretty clear that our autistic women responded much better to both of these group-based intervention programs. So the women in both groups are blue and red here. So we have MBSR women in blue, support education women in red, the men are purple and green. So for both physical health, women start out lower, which is one of the things that we talked about before, and both groups reported better physical health related quality of life at the end of their interventions. The men didn't change, but you could, call this a ceiling effect. For mental health, the groups are much more similar at baseline as far as their mental health struggles, but you really see the red and the blue bars come out as showing stronger average improvements in mental health, whereas the green and the purple kind of come out as a wash. I've seen autistic men's lives be changed by these types of things, so it's not everybody, but on average, women seem to respond better to this style of support. So then the pandemic happened. And you can't get people together and do mindfulness groups in the pandemic. So we switched to apps, and we wanted to know if those could be effective for autistic adults. And we wanted to try to address this persistent problem in the mindfulness research, which is after the study ends, people stop doing it, and then their depression or whatever their challenges comes back. So I worked with a new professor at ASU who's a behavioral economist and his whole program is about how to get people to form healthy habits. So from a very simple standpoint, we had one group that we told them, you're going to meditate when you leave the bathroom every morning. The other group, we told them, meditate 10 minutes a day. And then, of course, we had the waitlist control. So this is our three groups, app only on the left, app plus habit in the middle, and our waitlist control. Everybody went down in their depression, but only the app plus habit group was significant. And then when we asked them about meditating was when the really cool data came out. So this is how many days did you meditate on the y-axis and weeks on the x-axis. The red line at the top are the ones that we told them when to do it every day, the habit group. The dotted blue line are the ones that were just in the regular app group. The waitlist controls at the bottom. The line in the middle is when the 8-week intervention ended. And the red group kept meditating, and the blue group dropped right down to waitlist control levels. 6 months later, when we asked them about their depression, the red group here had maintained their depression 6 months later. The blue group, who did have a decline, but not significant, had creeped back up to about halfway to where they were before. So that's mental health. Got it covered. But how do we address persistent social and cognitive challenges in autistic adults across the lifespan? Almost all evidence-based social programs focus on young adults. So is it possible to adapt these strategies to be effective for middle-aged and older autistic adults? And then on the flip side, almost all sort of cognitive support programs focus on older and neurotypical adults. Is it possible to use some of those strategies to be helpful for autistic adults? And if we're going to engage in something like this, we need to be working with the autism community because we're really forging new territory. So that's what we did. So in a series of fairly well thought out steps, we started with focused interviews of autistic adults and a member of their support program about what are their challenges, what are they things that they think could be helpful or would not be helpful. We developed the content with autistic professionals and students giving their feedback. We delivered the content in a pilot study, and we included autistic guest facilitators and students in the delivery of the content. We collected the feasibility, acceptability, and qualitative data after each cohort, and we just kept making changes to try to address the things that people said we could do better. So we call this the Strengthening Skills program, where we're trying to address mental health, social challenges, and cognitive challenges all in the same program. For mental health, we're using mindfulness. For social, we're using a loosely adapted or an adapted peers version, which I'm sure most of you are very familiar with. And then for cognitive, it's a more loosely adapted program called HABIT. So HABIT comes from the Mayo Clinic, Arizona, and I'll talk a little bit more about that in a minute. So peers comes from right here, has efficacy with young adults, and has this weekly 16-week meeting, and involves a social coach that supports the autistic adult. Has strategies related to many of the social communication challenges that autistic young adults experience that I'm sure you're all familiar with. But young people are not the same as old people. So we were under no assumptions that we could just walk in with the young adult peers manual and that would resonate with 60-year-old autistic people. So we did a lot of modifications, some based on the structured interviews, some with our own insight, some as we went along, and people told us, I don't really like this that you're doing. Can you change it? A lot of changes were made around language. So even using the term social skills training can be very off-putting to some autistic adults. So we presented it as, this group is about deconstructing social interactions and sort of pulling the curtain away. It's like, what's going on behind the scenes when neurotypical people do and say the things that they do? And how can we make that more explicit so then you can choose what strategies you want to use? We're not telling you what to do, but we're giving you options so you can choose. Peers is very heavily focused on making new friends. Not every middle-aged and older autistic adult wants new friends. A lot of our participants had kids, and a lot of them, and that was not their main goal. Their main goal was, how do I get along better with people at work? How do I get along better with people in my family? Other program language, we really were sensitive to not having power dynamics in our, we didn't use social coach. We used program partner. We called it collaborative learning. The program, once the intervention started, the program partners were no different from the autistic adults. Everybody was working on these skills, which frankly, we all have room for improvement. We didn't use right and wrong or rules. Things were strategies or risky or something you could try. And we had to do some changes to the role play videos. Older autistic adults don't want to look at people in the hallways of their high school. And then something we found out very quickly is that not all middle-aged and older autistic adults have someone that can do a study like this with them, and they potentially need it the most. So we made the partner an optional aspect. And we also create a lot of space for autistic adults to share strategies that were working for them to make it this much more collaborative, intergenerational learning experience. So very quickly, HABIT, developed by Mayo Clinic to help individuals with mild cognitive impairment, strategies to preserve memory, improve quality of life, also a partner design, so that kind of melded well with peers. We really took our own adaptations as far as the topics we wanted to address, like making healthy habits, energy accounting, what are you actually up for today, what's reasonable, how to manage time. Other aspects of effective communication, autistic burnout became a big thing that people wanted to hear about, sleep hygiene, finances, all the regular stuff. So in our pilot study, we started with 14 participants in each group. We had the strengthening skills group that got it all, and then a group that got just the modified peers, because we felt like peers was going to work. We didn't know if it was going to work to throw everything together. And then the delayed treatment control, neuropsychologist, developmental psychologist, and mindfulness led each area respectively. We did this over Zoom because it was the pandemic, but I'm so glad we did, because now it's like an accessible, scalable thing that we can actually do. However, the standalone peers was an hour and a half. The three-prong was 3 hours. So we have worked with that a little bit to get the time down. Feasibility, fidelity of implementation looked great. Attrition rate was not too bad. We did have a couple more people drop from our strengthening skills than our peers, but it seems like we just got unlucky with COVID affecting our strengthening skills program. However, we have gotten the time down to 2 hours from 3 hours. In the acceptability questionnaire, which are typical questions, would you suggest this program? Was it effective? Did it help me? Rating from 1 to 7. Our strengthening skills participants were very close to a 7-point rating. Our peers participants a little bit below 6, and it is a statistically significant difference with the peers, or with the strengthening skills rating, that program higher than peers. I'm going to skip qualitative since I'm running low on time, but we can come back on this. The preliminary efficacy, we used the ABAS, the Adaptive Behavior Assessment Scale, and the World Health Organization Quality of Life Assessment and the subscales. Not really powered for repeated measures ANOVA, but we did it anyway, and we actually did find a significant effect on the ABAS social domain, which was pretty cool. What we were more interested in was comparing each intervention group to the delayed treatment control and looking at effect sizes. So when we do that, both groups show a large effect size on the ABAS social domain. The strengthening skills shows a large effect size on the hoopball psychological, which is the mental health domain. And then the strengthening skills showed a medium effect for the conceptual aspects of ABAS, which is more cognitive. So that's what we would expect from the cognitive aspects. Strengthening skills also showed a medium effect size for the quality of life related to social aspects. Overall, adaptive functioning showed a medium effect size for the strengthening skills, and then both groups showed a medium effect size for quality of life of your environment. So all in all, there's a lot more squares on the effect size, the notable effect sizes of the strengthening skills program. So we're very excited about that. So the takeaway is both of these programs can improve social functioning, but strengthening skills may be more beneficial for things related to the cognitive influences of your adaptive functioning and your mental health. And that's exactly what we expected from the 3 components. So very happy with those results. And we had people that were 20 to 70 all in a group together, and this suggests that can work. So yeah, feasible, acceptable, hopefully efficacious, and hopefully we can do a multi-site randomized trial with this in the near future. So my final conclusions are that seems like many autistic adults have persistent and or worsening challenges with their cognition, their mental health, and their social abilities as they grow older. Perhaps one day, if we keep chugging away at innovative brain imaging techniques and genetic techniques and a better understanding of sex differences, we could provide some precise care to autistic adults and their specific needs and what's going to work best for them. And we feel really excited about our multi-component support program that can address social, mental health, and cognitive challenges all in one to improve independence and quality of life across the lifespan. And this is my wonderful group of students and staff in a beautiful Arizona sky. And our incredible collaborators from a great Autism Center, SARC, TGen, Brain Imaging Center, Alzheimer's Center. We all work very closely together in Phoenix. Our participants are incredible and so dedicated to our research, and we're blessed with funding as well. Thank you.
Thank you.